Thank you. Namaste. Thank you, Surojit. Um, can we start the presentation? So, as Surojit said, last 30 years, I am in NCL, and my first tool which I used for chemoinformatics was Chemoxon. So, from 98, or you can see that I put the logo that 1998 it evolved. So, I'm using Chemoxon since then. Okay, and I kept some space. Whatever the new logo comes, I'm going to use it. Okay, so today I am going to talk about if at all you want to use some tool for handling large data. In the previous presentation, you must have come across that there is a data coming like a flow. Large data is coming like a tsunami of data. Now the question is how you are going to handle it. You are going to find one molecule, $1 billion molecule from that huge data. The faster, the better. If you want faster, then you need to use certain tools and then add some value to it. So that is what we are doing. So please feel cool, and this is not too much technical presentation. As a user, I am going to tell how I use the Chemoxon tools for developing my own tools, and then that will give you more confidence that this is the one tool with a lot of flexibility, freedom. So I will show you. So when it was free for academics, we started using it, and we continue to use it. Okay. I like to thank. So today, the agenda for my presentation is. People call molecular informatics, chemoinformatics, cheminformatics. I'm going to talk about how to handle huge data using the commercial tools and custom built tool. The second point is big data is there, how to handle big data. So you know that big data in biology, it is free, people can freely use, download gene data, protein data, etc. But in chemistry, it is now only coming after the PubChem. The lot of open data is coming in chemistry. Okay, the drug is a small molecule anyway. So then I will also talk about text mining scientific literature. This is where Chemoxon power you can see, name to structure, structure to name. These are all the very important component in Chemoxon tool, which we used for a long time, and we will continue to use it. Because the chemical can be, say for example, formaldehyde, it has 200 names. So which name you will use it for searching? So in that context, so this name to structure, structure to name is very, very important. Later, once you convert the name to structure, then you need to add some molecular features to that. That means molecular descriptor. Until unless you add feature to the molecule, you can't do machine learning, deep learning, prediction studies. So you start with literature, convert the name to structure, structure to data, data to add, then structure property relationship, then you can come to machine learning, deep learning, etc., AI technology. So I will, in the end, I will show you 2 years back, we got a huge funding from Government of India to design anti-cancer agents using AI. So I will just touch upon that, what we did. Because of COVID, it got little bit delayed in publication and release of the open source tools. So we use Chemoxons for academic research. I put it in, highlighted it. The best of Chemoxon tools and methods we are using for last 20 years. And some of the tools I listed, which I used frequently since long, that it will be very helpful for you. You can explore them. Okay? So now you know that Alexa is everywhere. So in future, if you want to tell Alexa, design an anti-cancer drug for colon cancer with these features, maybe one day it is possible. Maybe if you use Chemoxon tools, maybe you have your own tool which can use Chemoxon tools. This is what going to happen. People are making, like, I was so impressed to see Marvin Pro. So maybe the Marvin voice may come where you can speak the name, molecule will appear in the screen. Okay? So the future of AI. So there is a traditional way of monitoring the systems, now modern way of monitoring systems. So we don't know how much we are being monitored. So then traditionally the medicinal chemist used the molecular features, structure property relationship, if methoxy group is there, what is the expected effect in terms of activity, solubility, etc. If cyanide group is there, nitro group is there, what are the warheads, they used to do manually. Today, since the data is huge, it is humanly impossible to do such type of things. So what we used to do manually, now we are using computers to do the things, understand the features in a molecule and use it for prediction studies. That is what we are doing. So I think I spoke enough. So if someone tells me who she is, 1 million reward is waiting. You have seen this lady anywhere? Okay, 1 million reward. If you have seen, keep it with you. Tell me. Okay, so wait, I'm coming. You must have seen this person, again 1 million, another person, 1 million, another person, then there is no gender base, I put some person, 1 million, this is another person. So what did you see? You saw human beings. You see human being like people. Actually, this person does not exist.com. If you go and keep on clicking, you'll see the new face. Those people doesn't exist. Okay? What is the point here? The point is, when a AI can simulate a human being, which is, very, very complex, why not molecule, just atom, bond, some periodic table elements? So there is a possibility in future that AI is going to help you. So start using the tools. I started working on sustainable development goal. Then I saw that health is one of the component of sustainable development goal, and you can see this publication, I think people should see that the role of AI achieving sustainable development goal, and one of the component is health. Okay, so you can see that AI is coming everywhere. AI is in AI-based pathology predicts origin of cancers, and then you can see AI in machine learning for health. Then people use image processing for breast cancer analysis. So AI is coming everywhere. Okay, so why I'm telling this? We can't escape from this AI-based drug discovery. So it is better we educate ourselves, appreciate the technology, so that you will be leader one day in this field. So I entered the computer-based drug discovery program. When I was in master's degree, I was fascinated by Fourier reaction, Fourier reagent, etc. But if you see, he used computers in 1960s for computer-assisted organic synthesis. It was published in Science long ago. If you see the number, it is some 63 trillion number or something like that, way in which the carbon can form bond with to make new molecules. And eventually, of course, he got the Nobel Prize in chemistry. So the early adopter of computers, you can see that in chemistry, so he got the benefit. So I want to tell you, I'm sorry, you're all expert, but this is the workflow. Where we stand is, when we talk about in a drug, first identify the disease, identify the target, identify the molecule, then there is a process till it comes to the market. But when you talk about technologies, there are several technologies which used in drug discovery. Let us focus only at the molecular informatics level. So where there is a target, there is a small molecule, interaction, binding energy, and then you prioritize the molecule. So keeping this one in mind, so if you see that where the AI play important role, the AI play important role, these are the, I request the organizer to share my slides with the participants. So they, if you can't, you can see that, you can appreciate that from target selection, then compound screening and lead optimization. So this is where you need the tool. Compound is available, Chembl data is available, PubChem data is available, literature, SciFinder, anything you can find the data, whatever is published today. But how we are going to handle this? This is where you need to have a specific, specialized tools that can help you in screening. I will go into the details. So nowadays, people are publishing textbook, AI in drug discovery, etc. Advancing drug discovery via AI. So there are some, these books are published AI in drug discovery. So I don't want to go into details of this. One day, who knows, maybe we all join together to make another book, Practical AI for Drug Discovery. Okay. So this is the workflow. Let us, whenever you are talking about big data, the big data should have molecular informations and the bioactivity information. If you don't have these two components, you can't do drug discovery. Someone made the molecule, someone screened the molecule, the data comes to the public domain. Now you use the tools to learn that what is the variation in chemical structure responsible for variation in bioactivity. That is what in our AI domain we are trying to do. Okay, so these are all the things everyone knows. I will skip some of the slides. There are so many open source tools also available for conducting your AI-based drug discovery. These are the Chemaxon tools or similar type of tools can be complementing if you have some idea how to integrate them. Okay, so I'm listing this. This information is already available. So there are so many publications available with respect to open source data in AI. Okay. So now I will restrict my presentation only to the project which we did, that how to design an anti-cancer drug compound from the known data. So you know that cancer is very prevalent because of COVID. The impact or the importance of cancer is bit maybe slow down during these last 2-3 years, but then people are dying. So what I've shown here is the incidence of cancer across the world, but if you see that death rate is very high in underdeveloped country or poor countries compared to developed countries. So it is our responsibility to design the drug to save the people. And curing cancer is not easy, but what we can do as a informatics or drug discovery person, we need to identify the inhibitors with respect to pathway targets of interest. Okay, so I don't want to go into the details. So basically we are trying to identify anti-cancer agents, understanding how these inhibitors works with respect to specific type of cancers. Then there is targets, there is a pathway. Our job is to find the inhibitors, right? Okay, so treating cancer is also very complex. Okay, treating cancer is very complex, and our job is, say for example here I am showing rapamycin is a molecule, the small molecule which goes and interact with the different pathways so that you get the cure. I don't want to go more details into these things. As a drug discovery person, you must be more experienced and knowledgeable than me. Okay, then treatment of cancer is very painful. They remove the solid tumor, through surgery. They can use radiation. They can use chemotherapy. The problem is we have to identify a molecule which cures cancer at the same time not killing the normal cells. Okay, what is anti-cancer agent? Anti-cancer agent is to kill the cancer cells specifically without affecting the normal cell. So there is a challenge here. Okay, so then as a cheminformatics person, what we do? We identify all the anti-cancer agents available in public domain or FDA, drug data banks, etc. And we know that this molecule is for this particular type of cancer, and there is a particular mode of action. So can AI understand that this is the molecule, this is a class of molecule, this is the mode of action for those class of molecule for this type of particular specific type of cancer? Okay, this is what I am going to talk about. So I am coming from National Chemical Laboratory as a chemist. I was trained as a synthetic organic chemist. Then eventually I became a computer chemist. So my first, during my PhD itself, I was working on how to represent chemical structure. Representing chemical structure is very, very complex, and accidentally I opened, when I was making some documentation, accidentally opened WordStar. I don't know how many people will remember WordStar with molecular structures made by ISIS Draw or something of those years. I'm talking about 1992. Okay, then I saw that the molecules are represented as a matrix in the document. Then I started working how to represent chemical structures in matrix form. When you open in your software, it become a visual things. So that is how I developed interest in this field. Recently we published some work how to represent chemical structure as a barcode. So we know that money is represented as a barcode. Then why not, when I was seeing supermarket, people use this barcode for inventory management, etc. Then we thought that, okay, can we do it for molecules? Today we are able to put library of molecule in single barcode. Okay, this is also we filed some patents. Okay, so there is a possibility you can put a library of compound in barcode. Why? Just scan it, get some library of compound in your computer. Later you can use it for simulation, etc. Okay. So this is also very important because now people are talking about robot-based chemical synthesis. For that, we need to have automatic molecular structure recognition. So it's also coming. Where the knowledge is coming from when you talk about AI? AI to work, first you need to have a primary data. So this chemical knowledge is coming from several things. Can we harvest all the data, make a repository, use that repository for machine learning, deep learning to design a new molecule of interest? Okay, so here this slide I am showing, I borrowed from Chemoxon only. Today Chemoxon is capable of handling such a huge data. So this is a small number, maybe in future it will be 10x of these numbers. So you can see that when we talk about drug discovery, we need to handle such a huge big data. Maybe your $1 billion worth molecule may be hidden somewhere, or this will be the motivation to make a molecule of your interest. Okay, so this is actually, there is a data. From data we have to collect, make a information. From information we have to make a knowledge. So this is called inductive learning. So for that we need to have a molecule with bioactivity of interest in drug discovery context. But in some other context you may have, you know, polymer properties, you may have material property, etc. But molecule is very much important. Okay, so what AI does? AI does from given material, can you predict the property, or given property, can you simulate the molecule of interest? Okay, so again one more break. You see that these are all wonders of the world. What is the building block for this? Maybe some bricks or some marbles, etc. But when you talk about a drug, what is the building blocks for this? Okay, so natural product, people, when you talk about drug discovery, nature inspired drug discovery. People went to nature, got some bioactivity, now they try to find what is the chemical ingredient responsible for this, then they started doing synthesis. So this is the opportunity for you. The $1 billion molecule is hidden somewhere. Using the tools, can you make, using this tool, using etc., can you make that $1 billion molecule? I encourage people to go through that similar type of documents. And now you see that there are top 200 pharmaceuticals. See, in the actually money matters. So in this particular case, why these 200 pharmaceuticals are highly sold? Because there is a problem in the society. You could see that immunology, cardiovascular. So if you want to do drug discovery, you have to find out what is the problem, what is the solution, then you can really make an impact in drug discovery. Okay, so I just want to sensitize that when you want to do drug discovery, you need to know what is sold and why it is sold, what is the problem. With respect to that, if you do, there is an opportunity for you to contribute in all these fields today. Okay, then again I am coming back to my field, that is chemical structure and property relationship. At the end of the day, you have to correlate your variation in structure responsible for variation in biological activity, right? So now this is a very, very preliminary, very basic slide where molecule has an intrinsic property which you can calculate. Maybe using the Chemoxon calculator plugin, you can calculate all the molecular descriptors. And then there is an experimental way you can calculate the chemical properties, say for example, log P, solubility, etc. What is the use of the molecule if it is not soluble? So can you predict it? So this is also very important, and experimentally also you can measure all these things. I am going to talk that how we used these basic principles using some simple tools to reach this. So all the things also already published in one textbook, Practical Chemoinformatics. So there is a structure which you are supposed to handle, because that $1 billion molecule is a molecular structure. Now you have to predict the property or activity based on how you represent, how the computer understands to machine learning, deep learning. Finally, you have to predict the properties. For that, huge data needs to be processed. Okay, so now I also highlighted here your descriptor. Say for example, molecule, it is one of the descriptor. So you can calculate many descriptors, number of atoms present, topological descriptor, constitutional descriptor. There are several type of descriptors are there. So you can also, we used Chemoxon tools in the beginning to generate the basic descriptors. So I don't know how many people saw all possible calculator plugins that where you have API, you can generate, you can send the list of smiles, you get the Excel sheet or a matrix of a molecule with molecular descriptors. That is very, very important to correlate with your property or toxicity or activity of interest. So then the text mining helps you to find out who published what with respect to molecule. So humanly it is impossible for you to go through all the research papers. So this text mining also, we need to have a name to structure method available in Chemoxon, where you parse the abstract or full text, find the word, whether it's a chemical, whether it's a gene, whether it's a protein, etc. You recognize them and use this, make a network. So basically you can, using text mining, you can find disease, gene, protein, molecule, network. Once you make the network, you know what is the relationship, same disease, same gene, same protein, but different molecules. So that type of collection you can learn, and then which one is better, you can do it. So basically you try to understand, then you can design the molecule. So this is a standard structure activity relationship procedure. You take a molecule and then property, give it to the machine, then it does training set, test set, those days. Today the AI, you don't need to tell the AI, it understands automatically, okay? That is a new evolution happening. So we used these technologies, given list of molecule and activity, it creates the fingerprints, and then it, you can use substructure search algorithm, very powerful substructure search algorithm available in Chemoxon, where you can find the similarity. You can create a fingerprint, you can do fast searching, all the features are there here. We used all of them. Okay, so then we can generate the fingerprint for a class of compound. If you take a anti-cancer compound, what is the functional group means frequency of occurrences of functional group in a collection of anti-cancer agents. Among the anti-cancer agents, you can say that anti-cancer agent for colon cancer, anti-cancer agent for breast cancer, you can see this spectrum based on the frequency of occurrences. Now when you can use the spectrum itself as a filter to screen the library of compounds, whether that feature is there or not. If it is matching with this pattern, then it likely to be the anti-cancer agents. This is the technology we used for identifying the functional groups, identifying the substructure. We used the substructure algorithms available in Chemoxon. See, as a user group meeting, I have to tell that how we used it. Since I used it, succeeded to some extent, it will encourage you to try this. So now we are talking about chemophores. For a, suppose if you take anti-cancer agents, if you have some 500 functional groups, and then you can see frequency of occurrences of those functional group in the library of compound. Now if you want to use, you can also use this one like a Lipinski rule of 5 to filtering the new compound using this as a filter. Okay. After that, you can do advanced calculations, computing, docking, molecular dynamics studies, but the preliminary screening you can do rapidly using those tools. So you can do SOM analysis, you can find what are the eliminate. The library of compound can be seen as a single picture, so where each dot represent a molecule. So another 4 minutes or 3 minutes, I will try to tell that in the project what we did. We try to put everything together. So there is a Chemoxon engine in the backend, there is a data coming from public domain. As an academic person, I want to get all the knowledge and all the tools to solve some problems. So we used all these modules are there, which includes machine learning, deep learning, bioinformatics, genomics, etc., etc. Because cancer is a disease which is we need personalized medicine for that. So even TCGA, that cancer genome atlas data also needs to be integrated. So to make the story short, We took 3 million data points with respect to cancer, 10 type of cancer, we put into the system, then you can see that it's around 3 million data points. Then 10 types of cancer, then we subjected to neural network, machine learning techniques. We used deep learning and machine learning methods to build the several models. So this is the result, which is still under progress. So if when you see 0.9, 1.0, so it is still evolving. That's why we have not published it. It is still under progress. When you put the new data, the model changes, the prediction changes. So we used all these basic statistical methods. We also used RNN, LSTM, GRU, etc. for building the model. Okay, so since the data is huge, we need huge computing power, distributed computing systems to build the model and use those models. Now you give a molecule to the system, it will predict what is the activity, anti-cancer activity of that compound for 10 type of cancer using 10 type of methods. That is what the result would be. Okay, and still it is evolving. It will take some time for us to publish or something like that, because trust but verify, that is what we are working on. We want something which is reproducible, useful for the community. Okay, so now we understood what is the range of the bioactivity and what are the type of cancers and what are the molecular descriptors. Sometimes molecular descriptor may not capture the bioactivity relation. Okay, so now we have to, if some descriptor is not available, we have to add our own descriptor which actually relevant to bioactivity of interest. So 3 million experiment, went through machine learning, deep learning, then you build the model, apply the model on virtual library, and then you can shortlist the compound. So these are the component. Then at the end of the day, you need top 10 molecules with respect to the molecule of interest. So this is some screenshot of our program which handles a huge data, all type of cancer, all type of cell line data, protein data, gene data, even target data, etc., etc. Then we used the basic, in the beginning we used simple machine learning tools. Later we went to AI-based tools. Then basically you just put the list of smiles, it loads, and then you have huge computing resources. It distributes, it calculates, and it filters. Okay, so these are all the like progressive drug-like score, lead-like score, drug-like failure. All this information it automatically calculates. And in the back end, you can use a Chemaxon engine. Okay, you can do some visualization. You can, suppose you have a library of molecules against a list of targets, it does automatically in a distributed environment without any manual intervention. So we tested with the Veena and Veena-like tools. Now if you have a professional other tools, we can use it. So you can have a visual, we used JMOL for visualization, try to see the molecules and also the docked poses. Everything is automated here. Okay, so you have molecules, you have targets, then you use HPC to find the ligand receptor interactions, etc. So text mining workflow is shown here. So we published our molecule and property prediction. Then we published in 2005. It is one of the highly cited work. And this is melting point is very easy to predict. What about the other things? So I'm almost done. Another 2 minutes I will take. I'm another 2 more slides. Okay, so that was the 2005 work, and now this is 2020. So we used these type of tools to harvest the informations. Eventually it will be very useful for drug discovery. So what we are doing right now, we are also slightly moving to material discovery. We are working on little bit on polymer informatics, polymer with respect to environment, polymer with respect to medical context we are working, and we are also working on AI for chemical transformations. So why the reaction happened? So given a product, can you identify the reactant or the starting material of interest? Where is the variation in functional group? So we create our own fingerprinting, we create our own descriptor using Chemaxon tool as a substructure search algorithms to achieve this. So all the details which I presented is published in this set of articles, in the journal articles. We solved some problems, it was published as a journal article, and how to do it, we made it in the book format. You can see that things. Okay, this all the papers we published back to back. And this is very, very important take-home message for you. So sorry I put the old logo. I hope you will forgive me. And basically, these are the already the speakers spoke about it. We extensively used to achieve all these things what I spoke about. And thank you, Advent, for giving opportunity to speak my passion and hobby. Thank you. If you have any questions, please ask.
Thank you, sir. So if there are any questions, we can take them, or if there are none, please feel free to interact with sir.
I am
Dr.
here.
Dr. Suvendu has a question. Yeah.
Mike.
What's the difference between AI and the classical QSAR partial least square that we learned during our PhD days? I mean, is it like something like a old wine in a new bottle kind of a thing, or?
Actually,
Other
it is I don't want to compare wine and all these things. So we can say that supervised learning versus unsupervised learning. See, it is like when we make food at home in kitchen, it is a old QSAR method. Order Swiggy, Zomato is AI. Something like that. Tomorrow people want, Alexa, tell me my molecule. Alexa, design my anti-cancer, antiviral for this target. They want. So AI should avoid the painful way of finding the training set, test set, and then molecular diversity, variation in bioactivity, and the frequency of occurrences of molecule in the entire range. Manually we do it. Okay, so some they say, okay, only some set of 20 compounds they will take, make a QSAR, forgotten. Is it valid? Is it global? Is it applicable to future generation of molecules? No. So molecular diversity, understanding, selecting the compound, selecting the descriptor, selecting the method, these are all the new bottle. Thank you.
One more question related to that.
Good, I like that question. I love that question.
Now, when you talk about big data, how big should be the data? I mean, say if I have a data set of, for example, 100 compounds.
I can make 50 million compound for your 100 compound. I can make virtual library, I can generate from your 100 compound. I will discuss about it later. It depends upon what those 100 compounds. Is it only benzenoid compound, or it is a diverse molecule with 10 functional groups are there? So you have a library of place where you can connect, and what is the functional group you can connect. So given 100, identify, make a virtual Caesar to make a list of functional groups, list of scaffold, make 50 million molecules. Focused virtual library of 50 million compounds. That is big data. Think about 70 million compounds, 100 million compound available. Actually, I did this study in some 10 years back. Entire PubChem I took, 60 million compounds. I identified 6,000 scaffold, and then around 300,000 functional groups. I mean, it is not simple functional group. If you connect in a very benzenoid compound, so many types of functional group. So 300,000 functional group, some 600,000 scaffold. You can imagine, multiply by combination, how big it is. Okay, I'm sorry.
No, absolutely fine. Thank you, sir. Thank you for your time.
Thank you
for
so
your
much.
presentation.